So this is my cell signaling and communication project. The disease I chose was Alzheimer's with a nicotinic acetylcholine receptor. The receptor proteins that are involved in this pathway are G-coupled protein receptors, and there are two binding sites, and the binding sites are both on the extracellular domain and the cytoplasmic loops. The names of the ligands that are bonded to the receptor is nicotine and acetylcholine. Are second messengers involved? Yes, second messengers are involved. Are G proteins involved? Yes, they're involved. And they are alpha, gamma, and beta. Those are three examples. Are scaffold proteins involved? Yes. And are protein kinase involved? And the function of protein kinase is to activate signaling pathways and explain the steps of the abnormal signaling pathways. So what is Alzheimer's? Alzheimer's is a brain cell connection, and the cells themselves degenerate and die, and eventually destroying your memory and other important mental functions. So how it happens: first, Alzheimer's disease typically destroys neurons and the connections of the part of your brain involved in memory. Later, it goes to the cerebral cortex, responsible for language, reasoning, and social behavior. Eventually, many of the areas of your brain are damaged, and over time, a person gradually loses his or her ability to live and function. Ultimately, this disease is fatal. These are a few of the references I used to get my information. This is an example of the normal brain and what someone's brain looks like with Alzheimer's. This is also where the bonding sites are. So here you can see this one has 5. The extracellular domain, this is the more bonding sites as well. This is a picture of a healthy brain, someone with mild Alzheimer's, and then lastly, someone with severe Alzheimer's. The type of cell generated is nuclear. And there's a decrease in neurons from nicotinic acetylcholine receptors. And so there's a deficiency in the nicotinic acetylcholine receptors. And then how is the signal terminated? It is terminated because it is unable to bond with other proteins, so the signal could not carry anymore, so it just dies off. In Alzheimer's, the enzymes are inactivated that do not facilitate with nicotinic acetylcholine ligand and bind with the receptor, and also there's no degeneration of the receptor in neurons leading to the disease. And this is my concept map. So I used cell junction, and I defined cell junction, autocrine, puricrine, and endocrine signaling. And then I went from endocrine signaling and its long distance, then the three steps of cell signaling, and then it goes to the ligand, and then hormones travel through the body or the bloodstream. Then a ligand binds to the hormone, and then hormones are testosterone, estrogen, and those are just some examples of it. And that is my project.